enjoying the professional development events and the research talks that started today, we really encourage you to please go to blackandneuro.com where you can get a program for the rest of the events that are occurring until November 4th. And we want to say a big thank you to our sponsors who have made this week possible, and they're up on the screen here. I am incredibly excited to be introducing our first keynote of the inaugural Black in Neuro Conference. Dr. Gina Poe is a professor in the Department of Integrative Biology and Physiology and Psychiatry at the University of California, Los Angeles. Dr. Gina, she is a world-leading expert on sleep and its effects on memory and learning, hence my extra excitement for this talk. Dr. Poe was born and raised in Southern California by a high school educated single mother who believed education was the path to freedom. Oh, and there is her lovely face that has popped up. Hi, Gina. She earned her bachelor's degree in human biology from Stanford University. She then went to earn her PhD in neuroscience at UCLA in the laboratory of Dr. Ron Harper, studying neural mechanisms underlying sleep state. And during this time, she built and applied the first camera obtaining coherent images of subcortical neural activity in freely behaving animals. From there, she did her postdoc at the University of Arizona, and where she learned about multiple single unit electrophysiological recordings from freely behaving rats. And she actually sent those rats to space with NASA to investigate if the hippocampal mapping system adapted in zero gravity, which is super cool. At Washington State University, as an independent researcher, she won her first major research grant from the National Institute of Mental Health called REM Sleep and Memory, which is now in its 20th year of renewal. Dr. Poe continues her sleep and memory focused research at UCLA, while also directing several programs that mentor underrepresented students in the sciences. Dr. Poe's talk entitled The Neural Mechanisms Underlying Sleep's Unique Role in Memory, Insight, and Mental Health. Again, thank you so much, Dr. Poe, for joining us today, and we are very much looking forward to this talk.
Thank you so much for that wonderful introduction, Rekeb. This is really exciting to be here. I'm really excited to be here for the inaugural Black in Neuro conference. And I know a lot of you who have organized this, and I'm just so proud of you for doing this. It's wonderful to be here. I will share my screen now. And so let's see. Here we are. Instead of a title slide, I have just decided to start with acknowledgements because there are many people and things to acknowledge. These are the graduate students in my laboratory. Ward Pettibone and Raquel Guthrie are my newest members, and they are working on Sleep for Insight through the grant here that we just got for R21, Sleep for Insight. And Michelle Frazier is my senior most graduate student, and she's working on something that, a mystery that's been plaguing me for the last 15 years. How is it that the brain recognizes that something is familiar and therefore able to be erased once it's been consolidated from the hippocampus? And then Yesenia Cabrera is the graduate student who is doing the majority of the research that I'll be talking about today. So sleep-mediated healthy responses to stress and whether or not there are sex differences. This is the grant that I got while I was at Washington State University and have held on to, thankfully, to the generosity of the American public and this Institute of Mental Health, National Institute of Mental Health. And so it has funded almost everything that I'll be talking about today. So I was born in 1965. My father, Samuel Perkins, married my mother, who was a waitress. He was in the Marines, and both of them just had a high school education, but both of them believed firmly in the value of a good education and made sure that I got one. This is my father a little more recently with my brother, Tamir. And he's been a wonderful influence in my life, although I didn't get to see him very much when I was growing up because my father and mother divorced when I was 2. Every time I did see him, he was really supportive of me, told me that I could achieve anything that I wanted to do and that the world was my oyster, and I believed him. Here's him at my wedding. And he suffered, unfortunately, from undiagnosed bipolar disorder, which is one of the reasons my mother left him. She didn't know that he had bipolar disorder, but she did know that he acted erratically, and she was, not going to put up with that. So left him when I was really young, and my brother was really young. But this has probably been since I started neurosciences when I learned about his disorder. And this is one of the reasons why I'm interested in mental health. There's some really interesting and difficult nuts to crack left for all of us. This is my mother with my stepdad, a wonderful man. And my mother did raise me as a single parent mom with a high school education. She struggled a lot with poverty and sexism. And but she shielded my brother and I from all of the difficulties that brings, and made sure that we got a good education. So I ended up going to Stanford as an undergraduate, of course, on scholarship, major scholarships. I thought I would major in public health and hematology. I ended up being better at the hard sciences than I was at the public health side of things, but I was still bound and determined to do a PhD in public health. Unfortunately, at the time I graduated, there weren't any professional PhD programs in public health in the country, but Columbia told me, hang on just a couple years, we'll have one. We're starting one up. So I had 2 years to kill before my PhD. And I started as a waitress, and it just didn't work out very well. I always forgot people's orders. But my stepdad had a cousin who knew someone who needed a research tech at the VA hospital in Sepulveda. So I went to work there. He was doing a research project with F-16 pilots, test pilots at Edwards Air Force Base, to figure out what their EEG looked like when they were doing high G maneuvers, to see if we could tell whether or not they were going to go unconscious so that the Air Force could switch to autopilot. The test pilots never did badly, and certainly never lost consciousness, thankfully. So that was a bit of a bust, but we were able to switch it to a flight simulator and get a publication out of it by seeing when these test pilots were doing really well versus when they were struggling on the flight simulator, and we were able to tell something. So this is the first experience that I had with research, and the first chance I got to really contribute something real and lasting to the world, and that got me hooked. But what really got me hooked is I went to this first conference ever called the National Multi-Site Training Program for Basic Sleep Research. My PI, Barry Sturman, was invited because he was an early sleep researcher. And here I am back in 1988 with this group of amazing, wonderful, warm people. And they shared with us, the graduate students and the pre-graduate students, how little we knew about sleep and what the hypotheses were about why we slept. And I came to realize that these were the preeminent people in the entire world trying to figure this out, why we spent a third of our lives asleep and we didn't know why. So I realized that this is fun. This is fun to try and come up with hypotheses and ways to test them, to try and figure all this out. So everybody sleeps, all animals, from whales to worms and from dragons to Drosophila. Everybody sleeps, spend about a third of our lives doing it. Every animal that we've tested, even the ones where it's really, really dangerous to sleep, for example, the giraffes out in the wild, or if you're really, really sleep deprived, you can sleep almost anywhere. It's a really an imperative. The giraffe is the animal that has the least amount of sleep of mammals, only 4 hours per day, and only about 15 to 20 minutes of that are REM sleep, when you have atonia and all of your voluntary muscles are actively inhibited, so you don't act out your dreams. The giraffe has to lay that long neck out on the grass, and you can imagine out in the open like they are, that would be a very dangerous state. They are vulnerable to predation. All of our energy savings also seem to be minimal to nothing. We're not seeking a mate. Really, what are these essential functions of sleep? Here's a jellyfish which barely has a brain. It's not a central nervous system at all. And this is a jellyfish asleep in a tank. The researchers at Caltech disturbed it by doing a jet of water, and you can see it wakes up and tries to get away and pulses much more often when there is a jet of water. But eventually it goes back to sleep too. And if you do this a lot, it homeostatically regulates sleep. It gets more sleep after this sleep deprivation period is over. Interestingly, even this jellyfish seems to have what I consider two states of sleep. There's its pulse rate during the daytime, and at night its pulse rate slows down as it's asleep. And then there are even periods of time, 10, 20 seconds of time when it's not pulsing at all. And that to me is akin to that REM sleep period when you have atonia. So REM sleep also seems to be evolutionarily conserved. Here's Michael Chase, who organized that National Multi-Site Sleep Conference and talked me into applying to a PhD program in neuroscience. Again, I wasn't bound for that. I didn't think I was going to do it. But after that conference and after the experience of having something useful, I was hooked, and I applied to UCLA for my PhD program. This is the lab I ended up doing my PhD in. Ron Harper, Becky Harper, just warm and welcoming, wonderful mentor. And these are graduate students that I worked with. I really chose this lab not because of the research questions so much, but because the people were so great. And I knew if I was going to spend the next 5 or 6 years with somebody, I wanted to be with a group of people I really liked. David Rector was in my same class, and he was inventing a new way to assess neural activity. These were the posters, what they looked like in those days. You had to print them out and glue them to poster boards. And we worked side by side on the science and technology, pushing that forward. It was really a lot of fun. While I was at UCLA as a graduate student, John Lissman came and gave a talk, and he described a new finding that he and Patricio Huerta had found. In a hippocampal slice, if he added acetylcholine, he was able to cause this frequency of activity called theta, which is 5 to 10 hertz activity, which happens when in the presence of acetylcholine. And it happens in the freely behaving animal whenever they're learning. And when he was inducing this theta in the slice and then stimulated the slice at the peaks of this theta, when all the neurons are most depolarized, closest to their action potential threshold, he was able to get, with just 4 stimuli, really closely opposed, he was able to get long-term potentiation, which was thought to be the building block for learning and memory. So it's the thing that builds and strengthens synapses between neurons. Each one of these red dots is a synapse. You can see there are a lot of them. And really fascinatingly, he was also able to induce a reversal of that long-term potentiation when he stimulated at theta troughs, which was exciting because about that time, computational neuroscientists were hypothesizing that it would be really important to have depotentiation so that we didn't saturate all of our synapses and cause white noise to occur throughout the brain whenever stimulus came in. So especially important in the hippocampus because that's a kind of an assembly warehouse for memory. And from the hippocampus, memories get consolidated in a parallel distributed fashion to the cortex. And once that happens, you want to erase this small area of the hippocampus from that what was once a novel memory and now a consolidated memory, so the hippocampus can be refreshed and able to learn throughout the day. Unfortunately, up until that point, up until this finding, depotentiation was something that was also really not physiologically inducible. And so this paper showing that theta trough stimulation would do it was really exciting. It made me want to, when I got my PhD and moved to the University of Arizona, to really check out what was happening. In any animal that you can record the EEG from, you see there is beautiful theta that occurs when animal is running around and learning, and it also occurs in rapid eye movement sleep. Here is rapid eye movement sleep. Here's our sleep cycle throughout the night. The cycles of waking to N2 stage to N3, which is a deep slow wave sleep state, to N2 again, and then rapid eye movement sleep, which is where you get your dreams. In this N2 stage of sleep, you have what's called sleep spindles that occur once every 30 seconds or so, and they are 10 to 15 hertz electrical activity that comes from your brain, humans and other animals. And nobody really knew until 10 years ago what they might do. People kind of ignored this state. And in slow wave sleep, this deep slow wave sleep, this is the state in which we have the most unconsciousness. If you wake up someone out of slow wave sleep, which is hard to do, and ask them what they were thinking, they will say, nothing, I was asleep, let me go back. And they have a lot of sleep inertia and they will go right back to sleep. But in REM sleep is where you have your active dreaming state, which is where this theta occurs in the hippocampus. And the most vivid dreams. It really seems to be the state where decades of research have shown that it's really important for consolidating memories. So this REM sleep and this N2 transition to REM sleep state seem to be responsible for connecting and consolidating memories and removing the junk. I'm going to just walk you through for a moment what characteristics of sleep might be important to this memory consolidation and really what could go wrong in mental health disorders, especially those characterized by sleep disturbances. And almost all mental health disorders are characterized by sleep disturbances. Can these traits then be targets for therapeutic interventions? And really, what are the critical experiments remaining? And there are a lot of them. When I was going to do my postdoc at the University of Arizona, I knew they had this technique where you could record from a bunch of cells at the same time while an animal is freely behaving, running around on a track, and while it's sleeping again. So I wanted to learn this technique so that I could really answer the question of what is happening during REM sleep spontaneously in the freely behaving intact animal. If you record from enough cells in the hippocampus at the same time while an animal is running around on a track, you can see that different cells fire in different places, because not only is this hippocampus a memory assembly warehouse, it is also the place where we map our environment. So you can tell from which cells are firing and which are not at any given time where the animal is on the track. It's a really cool way to sort of read the rat's mind. And you can, again, read this rat's mind while it is asleep. So we did a task where I invented this task later when I was an independent investigator, where you actually bait 3 of 8 boxes around the track. The animal learns to ignore the boxes that smell like food but don't have anything in them when they open the doors. And then after they've learned that really well, we do a reversal task where they different boxes are baited. And so in the same environment, hopefully with the same map, they have to remap slightly to see where the food is now and start ignoring the boxes that used to be baited. This requires the hippocampus, both the mapping and the remapping. So again, going on this talk that I'd heard when I was a graduate student, I wanted to see what happens during theta, during wakefulness, and again during REM sleep, because that's the other time when you have a ton of theta activity in the hippocampus. Is sleep to remember, which would be what decades already of research had shown all the way since the early 1900s. They'd shown that if you don't get sleep after you learn something new, you will not remember it nearly as well. So REM sleep seemed to be to consolidate that memory. But then there was a new hypothesis about REM sleep for forgetting, maybe desaturating the brain. How would that happen? One of my hypotheses was that maybe if during REM sleep, neurons fired at that's when desaturation could occur. So I could directly test this hypothesis during my postdoc. And again, I found, as other people had, that the majority of bursts of activity, similar to the burst of activity that Lissman had induced, occur at theta peaks, again, when the neurons are most able to respond to that incoming input, and it causes long-term potentiation. So that's consistent. When a rat's running around, you get this beautiful theta activity in its EEG, and it stops when the rat stops to eat. And you get most of the spikes occurring at theta peaks. During REM sleep, what I found in the first data set I looked at was a majority of spikes occur at theta troughs in REM sleep, which was really exciting. This showed me that maybe REM sleep really is for desaturating the brain. Interestingly, during REM sleep, there's a different neurochemical environment. Here's during waking, you have all of these neurotransmitters on high levels. During non-REM slow wave sleep, they are waning levels. You don't have any acetylcholine. It's really important for long-term potentiation, and there's none of it during that deep slow wave sleep stage. But during REM sleep, you have lots of acetylcholine, lots of glutamate, which is the excitatory neurotransmitter workhorse that helps cause long-term potentiation, that building block for learning. But you have no norepinephrine or serotonin, which is similar to the slice environment that John Lissman had in his hippocampal slices. There was no inputs from the brainstem noradrenergic or serotonergic nuclei. So what might this allow uniquely during REM sleep? When norepinephrine is present during waking and during slow wave sleep, what it does is it occupies a receptor on the neural membrane that sets into motion a cascade of events that actually blocks depotentiation. That's what this slide is showing. It blocks depotentiation. So you actually cannot get any weakening of synapses, any clearance of the hippocampus or any other place when norepinephrine is present. And so the next data set I looked at was really cool. What I found is that even though during the learning session, day after day, the locus coeruleus is providing, the hippocampal pyramidal cells are firing at theta peaks consistent with long-term potentiation and learning. When the animals went into REM sleep after that learning session, only on the last few days of the week were cells firing at that theta trough, consistent with clearing the hippocampus of that memory. In the first few days, when the experience is novel, the neurons are firing at theta peaks again in REM sleep, as though in the first few days before the memory is consolidated out to the cortex, the hippocampus is preserving those memories by firing at theta peaks during REM sleep. And this was really cool because Michael Fanslow at UCLA had found that this period of time, about 7 days, was about as long as it was needed for memories to consolidate from the hippocampus to the neocortex, after which time those memories were no longer necessary. The hippocampus was no longer necessary to store those memories because it was consolidated out. So this was the brain showing me something that I hadn't even hypothesized. I thought that either the cells would fire at theta peaks or at theta troughs, but this shows me that there is a consolidation consistent timing and a switch. We're trying to figure out still what allows that theta peak to theta trough switch. But one of the things that's really cool is the locus coeruleus in the brainstem. Here's the rat brain, and the locus coeruleus way back in the brainstem provides norepinephrine all over the brain, including in the hippocampus. And the locus coeruleus stops firing in REM sleep. That's the one time it stops firing. It's consistently on, providing norepinephrine throughout wakefulness, slows down in slow wave sleep, and then stops in REM sleep. Here's the firing rate in the various stages of sleep. Intermediate sleep is another stage for another name for that N2 transition to REM, where you have those sleep spindles. So there's a very little norepinephrine during that time too. So one of the things that norepinephrine does when it's in the hippocampus is it allows, if there's acetylcholine, it allows there to be theta, but the theta amplitude is not as large as it is during REM sleep when there's no norepinephrine present. So if you, we infused norepinephrine into the hippocampus during sleep after learning. And we found that when we infused norepinephrine, it actually knocked the power of the theta rhythm down to the same level that it is during wakefulness. So here's saline infused versus norepinephrine infused during REM sleep. And you can see that when norepinephrine is infused, the size of theta is back down to the levels that you get in wakefulness. This is another experiment where we optogenetically stimulated the locus coeruleus to provide norepinephrine into the forebrain throughout sleep. And after learning, instead of a rise in theta power that you normally get during REM sleep after learning, that norepinephrine locus coeruleus stimulation knocks that theta power down to wakefulness levels again. So why is this important? Well, theta activity, which is normally large and steady during REM sleep, is really important for learning and memory. This is something that was found by Richard Boyce and Antoine Adamantis' lab. And what he found is that if he inhibited the ability of the hippocampus to produce that theta wave, he knocked theta down to about half amplitude, like norepinephrine does. He found that mice could not do a hippocampus-dependent task, a reversal task called novel object place recognition. In this task, you put two objects in an environment, have the mouse, check them out, and then 10 minutes or an hour or 24 hours later, you bring them back, moving one of these objects to a new place. And if the mouse realizes that something has changed and shifted, they check out the new object in the new place a lot more than they check out the old object, which is in the old place. That's familiar and boring. But if he inhibited theta during REM sleep in particular, he got animals that could not discriminate between the novel moved object and the old object. This is what the group is here, as compared to all kinds of controls that they had. They also couldn't do a contextual fear reversal learning task, which also requires the hippocampus. So what we did we did earlier in my lab is we tried to figure out how animals could do this reversal learning and whether REM sleep is really important for that. So we had them doing this reversal learning task where food was changed, the position of food was changed, so they had to stop checking out something that they had checked before. And what we found is that those non-REM spindles, where the noradrenergic levels are really low, and REM sleep all seem to be really important for consolidating and reconsolidating and integrating that new information into the map. So the less of that spindle state of sleep that they got, the less the new memory of where the food was integrated into their memory structure. And the less REM sleep that they got, the less they were able to remember where the familiar food was located when they did the familiar task every day. So the less REM sleep, the more confused they grew on that familiar maze. So it's as though that transition to REM sleep and REM sleep states are really for sorting out, what is true in what context. We did this study in using dizipramine, which is a selective noradrenergic reuptake inhibitor. It's an antidepressant that is sometimes used that leaves norepinephrine out in the synapse, acting at the synapse instead of being reabsorbed into the neurons. We did then another experiment where we optogenetically stimulated the locus coeruleus, which is here in the brainstem. It's a tiny, tiny area, hard to reach, but with the optogenetic stimulation methodology, we could actually stimulate that at any rate we wanted. So we stimulated at about 2 hertz throughout sleep, because 2 hertz is a frequency that we found didn't wake the animals up at all. It allowed sleep to be preserved. But did reliably invoke locus coeruleus activity. This is a response to blue light stimulation. You can see that there are action potentials in the locus coeruleus reliably with each stimulation. So you can see it's not changing the amounts of sleep-wake state. This is waking, slow wave sleep, REM sleep, and that intermediate stage of spindle sleep compared to animals that don't have that stimulation. But it really did reduce those locus coeruleus silences and reduces the stability of those place fields that I talked about. So when an animal maps the environment and runs around on that track, you can see that the same cell is firing in the same place lap after lap. These are 15 laps in that environment. But if we have during sleep after that initial experience, if we stimulate the locus coeruleus throughout sleep, you can see that the place cell activity is much less reliably firing in the same place each day. So you can see the correlation between activity place after place goes down if we've stimulated the locus coeruleus. Whereas about 50% or more of the cells in the control conditions have beautiful reliability and good correlations from lap to lap. After locus coeruleus stimulation for just 5 hours of sleep after an experience, you can see that they aren't able to reconsolidate, re-strengthen, and clarify where the place fields are. And instead, about 50% of them are firing with really low correlation rates. So this is the shift in the places that a place cell fires from lap to lap after that locus coeruleus stimulation during sleep. And it happens on the familiar task as well as the altered task where we move the food around. And you can see that day after day, even though we only stimulated the locus coeruleus during sleep, after days 1 and 2 of this dual task day, you can see that the animals that have their locus coeruleus stimulated they forgot where the food was on the familiar task, and they couldn't acquire where the food moved to on the altered task as compared to controls. It wasn't like we permanently lesioned the hippocampus, because the next week, if we took all the animals, didn't stimulate their locus coeruleus at all, and put them on a novel task in a novel place, all the animals were able to use their hippocampus and learn just fine. But somehow just during sleep, that disturbance of their sleep by locus coeruleus stimulation, keeping norepinephrine out there, was enough to make them give up and just kind of check every box as they went around. This locus coeruleus stimulation also disturbed these sleep spindles that I had talked about earlier. What normally happens is the locus coeruleus is off for the seconds prior to each sleep spindle in that transition REM sleep state. But when the spindle happens, then the locus coeruleus fires a rapid burst and ends the spindle. That's because norepinephrine provided by the locus coeruleus to the thalamus, which is the place where spindles are set up between the thalamus and the cortex, depolarizes the thalamus so much that they can't produce spindles anymore. So that constant stimulation of locus coeruleus throughout sleep really shortened this number of spindles, reduced the number of spindles and shortened them. This is what normally happens. The coeruleus discharge rate is really low just before each sleep spindle, and then goes to about 3 hertz during the spindle. And similarly, with our optogenetic stimulation, we were able to reduce them. The spindle state is really cool. Like I said, about 10 years ago, people started being interested in it because they found that memory consolidation seemed to be really positively influenced by spindles. This is a study from Siapas's lab at Caltech that shows that the hippocampus and the cortex are uniquely in a kind of coordinated they're communicating really well during sleep spindles in a way that they're not even communicating during REM sleep or during that deep slow wave state of sleep. So when the hippocampus fires, the cortex reverberates in a spindle specific frequency and fashion. So those brief moments of 1 second to 2 seconds long of the sleep spindle seems to be a time when the hippocampus is communicating out to the cortex, and it might be a time when those new memories are being consolidated out there and reformatting our memories. The other time the locus coeruleus fires during the slow oscillations of slow wave sleep is prior to every most depolarized state of the slow oscillation. And it's really cool because these slow oscillations is where a lot of the place fields, the place cells are reactivating. A lot of cells are active at these peaks. So the locus coeruleus fires normally during slow wave sleep to allow norepinephrine to be present at the peaks of those stations. And what I think that that is doing is it's preserving all of those memories that are not being reactivated. So what happens when one circuit is being activated and another circuit that might be encoded by some of the same cells is not activated, in the absence of norepinephrine, you can get heterosynaptic depotentiation, a erasure of those neurons, of those synapses. But when the locus coeruleus is firing and provides norepinephrine, you can't get depotentiation, you can preserve all those orthogonally encoded memories. And so I think really what the function of the locus coeruleus firing during slow wave sleep, when there's a lot of reactivation, is to help preserve our memories so that they aren't slowly erased over time. It's interesting because in Alzheimer's disease, the locus coeruleus starts misfiring. That's one of the first things that happens in Alzheimer's disease is the locus coeruleus starts misfiring. It messes up these deep slow waves and probably messes up the preservation of memories that is allowed by the locus coeruleus during sleep. So instead, sleep becomes a maladaptive time when our memories can start to be erased. Okay, so I talked about how depotentiation was important to not saturate our hippocampus so that we can continue learning throughout the day. It really turns out that seamless learning throughout our lifetime really requires this circuit remodeling. Where is the food now versus where it was before? You have to sort of change your mind. You have to reformulate your schema of the world every time you learn something new and need to fit it in. And that requires depotentiation. So for example, if you're in the theater of war and you associate helicopters, the sight and approach of it, with the fact that it might be spewing bullets and you need to duck and run and get out of the way, well, that's really an important and learning task that happens during the daytime and gets consolidated at night. But once you come home into a different context, hopefully not the theater of war anymore, you have to reassociate the sight and sound and approach of a helicopter with something new, which at home was probably something safe, like the news, and maybe it's just a news helicopter. So you no longer want to duck and run and get out of the way once you're in the context of home. And if you can't do that, if you can't reformulate your schema in the context of home, you could confuse the old fact, which is that it's a fighter helicopter, with the new fact that it's probably the news, and not really know what to do. Like our rats were confused when we moved their food and we kept norepinephrine present in the hippocampus throughout sleep. If we can't do that depotentiation, that weakening of the old fact with the schema, then we won't know what to do. And this is probably what people with PTSD suffer from. Fear extinction memory is really a type of reversal learning, and PTSD patients really have a key deficit in this reversal of fear. So one of the ways we can model this in an animal is we can give them, fear conditioning. And we basically take a particular context, which has a particular smell and sight and lighting characteristic, and give them a few shocks, 5 tones paired with shocks. And after a time of that, they'll start freezing in that environment. That's one of the fear responses, is freezing. If a day later you take them into a different environment and give them the same tones, initially they're going to freeze a lot, but with 30 of them, if you never shock them, they will learn that in this new context, they're not going to be shocked, like coming home from the theater of war. Their helicopter is not going to be associated with bullets. If the next day you bring them in that same safe context and test them by giving them some more tones, you can see whether or not they were able to consolidate that new memory into their old schema, that in this context, those tones are no longer associated with anything fearful. So this is research that is being done in the lab by my graduate student, Senia Cabrera. And what she's doing, she's recording the locus coeruleus throughout sleep in males and females after a stressful, traumatic learning event, and seeing if they can reverse fear after this. So we want to find out, does REM become abnormal, and does that abnormal REM really become maladaptive to animals? Can they not reverse their fear anymore? So it turns out that people with PTSD, as compared to people who are exposed to similar traumas but don't have post-traumatic stress disorder, people with PTSD have really low REM theta power. So that theta power I was talking about before is really lower. And that's consistent with what I was talking about with too much norepinephrine in the brain. And there are lots of other pieces of evidence, indirect pieces of evidence from people with PTSD, that the norepinephrine levels don't ever go down in REM sleep. So there's really no silence of the locus coeruleus probably in humans, and certainly we are finding in rats with PTSD symptoms. So I'll show you some examples of that. We started doing this research in 2016, 2015, and we are continuing this. Seems to be especially true in women. But I'm doing this research. I'm really excited that my research has led to this, because it turns out that several of my family members do suffer from PTSD. This is my stepdad, who was in the Navy for 20 years. He fought in Vietnam, he fought in Korea, many, many tours of duty, and lots of facing enemy, and lots of decisions that he had to make as what he became eventually a commander of the ship. And he has PTSD today. It's been many, many years since those wars, but he still suffers from post-traumatic stress disorder. I'm also excited because my brother, with whom I was raised, came here. Here is back in his innocent days. He also joined the Marines and saw active duty in the theater of war, and also, unfortunately, got PTSD from that experience. So it's really cool, because the research that I get to do today relates to that. I just started out in the function of REM sleep for learning and memory, but it's led me to this PTSD related research. And finally, another dearly beloved family member. This is my mother's brother, her younger brother, Danny. He fought in Vietnam. And when he came back, it was clear that he also suffered from PTSD, and a few years later committed suicide. He took his own life. He was, of my mother's brothers, the one who looked after my brother and I most. He brought us bikes and taught us how to ride them. He played his flute for us. He had a beautiful daughter at the time too. And when he committed suicide from his PTSD, which happens to 22 service members still to this day, every day, he left behind his family who adored him and who mourn his death all the time. So there's a really high cost to post-traumatic stress disorder. So what we're looking at in my lab is what happens and how can we cure this? Normally the locus coeruleus doesn't fire much at all during REM sleep. But if you expose animals to a trauma, this is the worst day in their lives. We bind them for 2 hours like they're in the claws of an eagle. We put them in a tub of water swimming together. It's not too cold, but they don't like not being able to get out. And so they're dunking one another, trying to climb over each other to get out. It's very stressful, 20 minutes, very tiring. We towel them off. We put them to rest, let them rest for 15 minutes. And then we put them in a bell jar with ether. Ether is a direct activator of the stress axis in our brains. So they only have 5 minutes of this exposure, but it's a third sort of insult to their whole stress systems. And then we kind of add insult to injury and isolate them in cages for an entire week after that. And that is enough to cause the majority of the animals to get a PTSD-like phenotype, which is failure to extinguish fear or consolidate that extinction fear that I talked about earlier. And what we found in some preliminary data is that the locus coeruleus right after that traumatic experience, well, it's firing a lot more than it does normally, and it continues to fire in REM sleep. It fires again during REM sleep, when a time when it should normally be off. So here's their animal's acquisition of that. A week after that trauma experience, we put them into a fear conditioning chamber and let them associate that chamber with and that tone with shock. And you can see them starting to freeze after about 2 or 3 of those tone shock pairings. The next day, we give them all extinction training, those animals that haven't gone through that single prolonged stress, and controls that had never been stressed before. And you can see all of these animals are learning to that this environment, this new environment, is no longer associated with shock, so they stop freezing after a while. But the next day, when you bring them back into that safe environment, you can see the animals that went through that traumatic experience aren't able to consolidate and recall that in this new environment, that tone is not associated with a shock. So you can see they have an extinction recall deficit, much like people with PTSD. And what happens to their REM sleep? Well, I showed you before that the locus coeruleus continues to fire. Here's the locus coeruleus activity as recorded in my lab. You also get much more muscle activity during REM sleep. Normally our muscles are flat and we don't act out our dreams, but I've seen a rat after this traumatic experience during REM sleep wake themselves up by throwing themselves across the cage because their myoclonic jerk, that's the activity of the brain overriding that muscle inhibition, was so strong it threw them across the cage and they woke themselves up. So there's a lot more. And people with PTSD also have much more active REM and wake themselves up a lot more out of that REM sleep state. So the animals that had the most difficulty with freezing are also the ones that had the most REM sleep after trauma. So they had the most of this, what we are calling maladaptive sleep, with the locus coeruleus active right after that trauma. We also have another model for trauma called stress-enhanced fear learning. And that was invented by Michael Fanslow at UCLA. And we also see that animals have a lot more REM sleep after that traumatic experience. And we also see in our lab that the theta power during REM sleep is much lower, consistent with that increased activity of the locus coeruleus, more norepinephrine in the hippocampus. And that power, the reduction in REM theta power, is also correlated with how much freezing these animals do after that fear extinction learning. So are the locus coeruleus firing rates elevated post-trauma? I showed you this one slide showing you that, yes, there is more firing after trauma in REM sleep. 4 days later, there's still more firing in the locus coeruleus. So this is a long-lasting increase in the firing rate of the locus coeruleus during REM sleep. Yesenia is looking at this in females as well. Sorry, you can't see this very well, but this is the firing rate of females actually not even exposed to a trauma. This is during their low hormone phases of their estrus cycle. You can see that in females, she's the very first one to record from the locus coeruleus in females, she and a postdoc that worked with us, Chia Chen. And you can see that even in animals that have never been exposed to a stressor, during the low hormonal phases of their estrus cycle, their locus coeruleus isn't shutting off. And this might be one reason why females are 2 to 4 times more susceptible to PTSD than are males. If the trauma comes at low hormonal phases of their estrus cycle, you can imagine that, that their locus coeruleus is not able to shut off like it would normally. So here's just a complicated model. We published it in the Journal of Women's Health, so you don't have to memorize it right now. But normally the takeaway from this is that if you get good sleep before your trauma and the locus coeruleus is your hippocampus is not saturated. It can encode all the context and cues. The locus coeruleus is able to help your brain learn during wakefulness, and learning shuts off the locus coeruleus activity. Then your REM sleep can be normal with no locus coeruleus activity, and it can consolidate what's important. It can erase what's not important from that learning experience. And when you go through extinction learning, or like exposure therapy in humans, you can encode all of the associated cues and extinguish that fear, because in your new context, your hippocampus is free to learn those new context cues and able to help you learn that. So this is not longer a place to be afraid. But if you have poor sleep and you get more REM sleep after your trauma, and your hippocampus is if you get poor sleep before your trauma, is like you can imagine in the field of war, the theater of war, you might not get great sleep before a big battle. Your hippocampus would not be as desaturated by that poor sleep. And so you aren't able to encode all the important cues, and the locus coeruleus isn't shut down. And during REM sleep, and your hippocampus really gets saturated with that traumatic memory. It only serves to, reactivation only serves to re-strengthen those traumatic memories. And so your hippocampus is saturated, and extinction exposure really doesn't work. And that sort of continues as a vicious cycle: poor sleep, locus coeruleus active, and unable to encode those safety cues. You can imagine too early of REM, increased REM, too early when your locus coeruleus is still active, might switch you from a healthy cycle to an unhealthy cycle. And also low estrogen levels, when your locus coeruleus is already firing during REM sleep anyway, might switch you from a healthy sleep response to an unhealthy sleep, a maladaptive sleep response. So some therapeutic approaches one could try is you could try manipulating REM sleep, maybe preventing REM sleep right after trauma until your locus coeruleus is calm. You could try blocking the beta adrenergic receptors in the brain during REM sleep right after a trauma too, or after extinction learning. You could try enhancing the adaptive replay activity during sleep. You could try monitoring people's locus coeruleus activity through non-invasive methods, and see when their locus coeruleus is calm enough, then you allow them to go to REM sleep. If it's not, you give them sort of self-calming tasks to do, so that when they do go to sleep, the locus coeruleus is nice and quiet. So we're testing some of these things in the lab. We're using propranolol, which is a beta blocker, giving it to animals after that fear extinction learning event, and seeing if that really helps with extinction recall if they've been through a traumatic event before. And in fact, early propranolol given just before the animals go to sleep really does knock their freezing down to control levels, animals that have never been through a trauma. This is preliminary data, but it's really promising. All right, so we can also REM sleep deprive them and see if that works right after the trauma. And in fact, that also prevents most of the animals from getting the PTSD phenotype. So we're really excited by this. This is the work that is being done on PTSD in the lab. Yesenia Cabrera I mentioned before. Jasmine Holloway is a research tech that just graduated from UCLA, and she's doing a lot of this great work with propranolol. And Gabriel Rojas-Bo is a undergraduate student from the University of Puerto Rico, and he was able to do a virtual experience with us this past summer to test whether REM sleep deprivation after a traumatic exposure will really help rescue animals from getting PTSD. And here are some of my collaborators, my former postdoc, Willie Vander Hayden, Chia Chen, who with the first from females, and Israel Lieberzone, who developed the single prolonged stress method of causing a traumatic experience in rats. This is my lab today. Many of the members, we have a great time. The same reason I chose to be a neuroscientist, because I loved the community, is what I love about my lab these days. Here we're wrapping Christmas for students. Also, I wanted to let you know, Black and Neuro, that there's an open rank tenure track job position at UCLA in the life sciences for those who have a real heart for mentoring students from underrepresented groups, as UCLA has. 25% of the student body are underrepresented in the STEM fields. And so we're hiring someone who has a great track record, a great research program, and also would be a great mentor for our students. So if you want to apply for this, there's 10 more days that this job is open. This job position is open for applications, and I encourage you to apply. This is a picture of some female neuroscientists at the Society for Neuroscience meeting in 2019. And I just want to say thank you to my lab, thank you to neuroscience, thank you Black in Neuro for having me as a keynote speaker. And with that, I'll just leave up here some really critical unanswered questions that we need to address. So thank you for your attention.
Thank you so much, Dr. Poe, for that incredible talk. Thank you for sharing not only your work and your journey to research with us, but importantly, your lived experience and that of your families. That, of course, is very much embedded and connected to the work that you do. So thank you so much. We have a few questions for you. So I'm going to start off by asking one of them. And for all of our attendees, you are still able to post your questions in the Q&A if you have them. We'll try to get through as many as possible. So
I'm
the
going to
first
stop
question,
sharing if you don't mind, if I can get there.
Yes, absolutely.
Yeah, okay. If I can find the stop share button, it's hiding underneath my menu here. Can you possibly stop the share for me? I don't know if that's possible.
Ooh, I do not know if that's possible. I do not think it is.
Okay. I'm sure I can find it.
It usually is a little tricky.
Yeah.
Yeah.
Okay, I found it. I found it. Good.
Wonderful. Okay. So the first question that we have is from Memudu. Hopefully I said your name correctly. Since the hippocampus is responsible for memory, what happens when one cannot recall a dream when awake from sleep?
Oh, that's actually a good thing. So the locus coeruleus, like I said, normally is off during REM sleep, and that's a good thing. So it doesn't help us, doesn't push things towards long-term potentiation. It helps us to have these free dreams without having to remember all of them. It's fun to remember dreams, I think, in general, except when they're nightmares. So it's not until we wake up. It's kind of like our working memory is working while we're dreaming, but our ability to transfer that to long-term memory, which is helped by the locus coeruleus, norepinephrine, is gone. So if you wake up right from a dream, our locus coeruleus starts firing, and whatever's in our working memory can get potentiated, and we can remember that dream if we rehearse it right away and think about it right away. But if we don't, just like anything out of working memory, it just fades away. I think it's a good thing.
Yeah. And our other question here is from Tyler, who asks, these sleep-related disruptions impair hippocampal dependent behavior. Is that because they are playing a role in the computations that the hippocampus supports, or because they are disrupting other functions like metabolic functions of the hippocampus, which then have an indirect consequence for the hippocampus dependent behaviors?
Yeah, I think both are true, actually. So sleep, all of the states of sleep are important. And I didn't talk about it, but during slow wave sleep, proteins are being built, and you need proteins built to consolidate memories. That's been shown quite clearly by a lot of different studies. So proteins are being built at uniquely high rates during slow wave sleep. So if you have slow wave sleep disturbances, or you don't get enough slow wave sleep early enough in the consolidation phase, you can actually inhibit your ability to learn and remember. and consolidate those memories. And then during REM sleep, like I told you before, your memories are being strengthened if they're new, so they're not erased and they are preserved. And the memories that need to be erased, that have been consolidated already, are getting erased. What I didn't really talk about is during sleep spindles, I did talk about the unique communication between the hippocampus and cortex, which occurs against a backdrop of silence of the locus coeruleus. So you can reformulate, weaken the things that need to be weakened, strengthen the new things that need to be strengthened. People with schizophrenia actually have very few sleep spindles, and they're very short and very small. So that integration of new information is probably can't really happen very efficiently in their transition to REM N2 stage spindle sleep.
That's really interesting, because in the research that we do as well, we're finding very atypical characteristics of sleep spindles in children with intellectual disabilities and autism as well.
Wow, really neat. I want to see those publications.
Absolutely. So our next question from an anonymous attendee is there any correlation between the increase in LC firing and the level of stress or trauma the animal experiences?
Yeah, and I think that's a great question. I think we don't know the answer to that yet, but we haven't done enough rats to be able to see a correlation. But there are lots of individual differences in our responses to stress, right? And interestingly, estrogen has a really big effect as well. So the locus coeruleus is covered with estrogen receptors, and with opiate receptors, and both of those really influence how the locus coeruleus responds and how long it responds to a stressor. So different people have different, neurotransmitter and receptor levels on their locus coeruleus. So I would imagine that different people and different times of our cycle, we have different lengths of time that we respond to a stressor, and that could affect our sleep depending on how long it lasts. So great question. We need future research to really answer it.
Great. Our next question is from Dory, who asks or says, firstly, thanks for the amazing talk.
Well, thank you for attention.
Does the LC activity also affect replay and or pre-play activity in the hippocampus during learning?
Also, we don't know the answer to that. We do know that during those slow wave, sharp wave ripples, slow activity, it is on just before the peaks when replay happens. So it would be really cool to silence the locus coeruleus whenever it would fire just prior to those slow waves and that replay, and see what that does to memory. And I would imagine that very selective, very temporally specific silencing would be enough to disrupt memory consolidation. But nobody's done that yet. We just now have the tools to do it. Yes, you guys can do it. Please do.
So we have a question from Borla, who again says, wonderful talk and story. Thank you for putting this Black in Neuro conference. Oh, thank you for joining. Do you think the opioid system could be used to improve sleep and to treat PTSD via the LC?
Ooh, wow, that is a great question. Probably on a very selective basis. Be careful, because we know that long-term opiate use, what that will do is it will down-regulate the number of opiate receptors on the locus coeruleus. So the locus coeruleus becomes even more, in the absence of opiates, then when you withdraw from an opiate, your locus coeruleus becomes even more hyperactive. So I would imagine that perhaps a single use before sleep, after a traumatic event, might really be helpful. What I don't know, though, is what opiates do to non-REM sleep, because all of it's important. So in fact, actually, opiates do suppress REM sleep altogether. So it doesn't allow REM sleep, but it doesn't allow it to be its adaptive self either. And good adaptive REM sleep would help us to figure out what's important from what we've learned and what's not important, and contextualize our fear memories. So you would imagine good REM sleep is really important to have. And if opiates completely suppress REM sleep, as they do, you don't want that either. So, but it's a question, and it would be interesting to see what the results would be.
I think we're just formulating multiple PhD research questions.
I think so. Really important ones.
Important ones, yes. So we have a question from Barbara, who asks, are the trauma-induced memory and extinction deficits specific to aversive learning? Would the rats that experience the trauma or stress of swim or isolation also have deficits in appetitive Pavlovian conditioning?
Yes. Israel Liebezon really looked at this and really fleshed out the model of single prolonged stress for PTSD. And there were a lot of other things that are PTSD consistent, phenotype consistent with this traumatic exposure. One of the things that we found, and also other people in Israel's lab found, is that reversal learning of all types is impaired after this phenotype is developed. So even positive appetitive kind of reversal learning, for example, on our 8-box maze, is impaired. So it's not just fear extinction, it's all reverse. If you saturate your hippocampus, it becomes unable to do what it's supposed to do. And so reversal learning is impaired, yes. Israel did publish a couple of papers on that. We have some papers, some studies from our 8-box maze that we have not yet published, but need to work on that and get that out there.
Okay, thank you. Our next question from Heather asks, the administration of the beta blockers, how do you discern possible effects on motor abilities, I'm assuming, as it is often used for tremor and effects of peripheral beta receptors?
Okay, yeah, and I'm so glad you asked about the beta blockers, because there have actually been 8 clinical trials of fear memory and beta blockers in humans. 4 of them worked beautifully and 4 of them did not work at all. And I looked closely at these studies, and the ones that didn't work, one was a dose that was too small. It was actually a low dose of propranolol that is given to people with heart conditions. So propranolol is commonly prescribed for cardiovascular disease, and but the dose is fairly low, and it didn't work in people at that low dose. You needed a 40 milligram per kilogram dose. In rats, we tried a low dose, it didn't work either, and in people. The other thing that none of these 8 trials in humans took into account was sleep. So if they gave propranolol, if they did the reversal extinction learning in the morning and gave propranolol right afterward in the morning, and people didn't sleep, propranolol wears off after 6 to 8 hours. And so if then they sleep that night, then their locus coeruleus could be active again without that propranolol protection. So really calls for, this research in rats really calls for a revisitation of those propranolol experiments, but keeping sleep in mind, so that propranolol is active during sleep after that trial. So it's my hypothesis that the 4 trials that worked really well, even a single dose of propranolol was able to completely extinguish the fear responses, allowed their patients or their subjects to go home, take the nap. Propranolol would make you, at such a high dose, would make you sleepy. And so I hypothesized that they did go home and take a nap. And that's why it worked.
Great, thank you so much. And so another question that we have here for you is the extinguishing, for example, freezing, that synaptic pruning occurring, or do the connections remain for potential spontaneous recovery?
Oh, that's a great question too. So depotentiation really is just the first step in the cascade of events that would eventually retract synapses. So would imagine that if you've depotentiated and don't reestablish that, and maybe it takes a week. I don't know how long it takes for a synapse to be pruned away. I would actually, in some mice studies with 2-photon imaging, it looks like pruning can happen right away. But there's something else that needs to happen. Is not only does the axon need to retract, but the extracellular matrix needs to reformulate to not allow that sort of that tunneled path between presynaptic and postsynaptic side. There is a kind of a new idea about how long-term potentiation works, that memories are really expressed through the synaptic connections, the strengthened synaptic connections, but they're retained in the genome and the epigenetic changes in the genome. So that, for example, a hibernating animal that has about 90% retraction of axons while they're hibernating, when they wake up from that hibernation, they actually can remember where they had food before. And and we don't know whether or not the axons grew down the same tunnel, the extracellular matrix tunnel, or whether there are new synapses formed between the presynaptic and postsynaptic side that didn't exist before due to the epigenetics changes that might occur both presynaptically and postsynaptically. So there is a lot of questions left to answer, but so depotentiation is just the first step. We'll put it that way.
Okay.
These are great questions, by the way. Wow, you guys, and these are the best questions that I've ever gotten from this talk. So I appreciate you guys.
High praise, high praise. Thank you so much, everyone, for submitting your questions. I will ask one or maybe two more, and then we're going to have to start prepping for our next data session or our next speakers. Okay. So yes. We have one from Lauren here, who says, thank you for such a beautiful talk, Dr. Poe. Are you aware of interventions that could align the timing of LC firing during sleep spindle oscillations in an effort to mitigate the negative impact of misfiring in Alzheimer's? Ooh. And so she points to methodologies like acoustic stimulation and pink noise having emerged to entrain sleep spindles and slow oscillations during sleep.
Yeah, this is a fantastic question. Again, this is a really new field, and these interventions are really cool. And you can elicit spindles with acoustic stimulation. What I don't know is if those spindles are normal in the sense that they're preceded by silence of the locus coeruleus, which you would need to be able to reformulate the neocortex, and whether or not they are ended by locus coeruleus spontaneous bursting. It's been difficult historically to record from the locus coeruleus. It's deep in the brainstem, and it's really tiny. There aren't very many labs in the world that are doing it, especially from a freely behaving animal. So, so we don't know the answer to this yet, but it would be another great dissertation topic. There are some really great methodologies allowing us to record from a bunch of locus coeruleus neurons at the same time, including mini scopes. So, or what's it called? I don't remember what it's called, photovoltammetry or something.
Definitely not my field, so I cannot comment on that. But if anyone knows, please feel free to put it in the chat.
Yeah. You can use tungsten electrodes, and we use tetrodes in our rats. So it works. So,
just going through the chat, we're seeing so many people thanking you, wanting to actually, they're interested in doing more sleep research. One of the questions is, are you looking for a PhD student? I can't remember who said it because I've scrolled past it, but
That's all right.
They're interested in doing some research with you, Dr. Poe.
Well, yes, of course. My senior-most graduate students will only be here a couple more years. And if you apply to UCLA's neuroscience program or the MIP physiology program, or several other programs, you can do a PhD with me because I'm affiliated with all of that. At UCLA, we do rotations in first year, so everybody does 3 lab and make sure that there's a good fit. So if you apply today to UCLA, then it's possible that 2 years from now, you could be in my lab starting your dissertation research. I don't know what my funding will be like. I know I'll still have my R01 at that point. I don't know if I'll have a new R01 for the PTSD research. I'm thinking that there will be an R01 or some other funding mechanism that will fund it. So yeah, absolutely. I welcome you. It would be great. I might need a slightly bigger lab if I need to extend it, but we can negotiate that.
Well, I think, yeah, everyone look forward to that in 2 years, marketing your calendars. And again, I mean, there are so many questions still from the community. What we'll do is we'll collect them and we can send them over to you, Gina, if you have the time to answer them. So thank you.
Oh, I'd love to.
Great. Thank you, everyone, for tuning in and for sending in your questions. And most importantly, thank you, Gina, for this incredible talk. And we look forward to seeing you again. And to the rest of the viewers, please come back in about 10 minutes. We will be hosting our data blitzes and our talks. So we've got more cool research from the Black and Neuro community to come. So thank you again, everyone. Yeah, see you soon. Thank you, Gina.